Oh yeah.
And so I wanted to see if you
It's fairly like 11:30.
Oh, I love it.
We're pretty much done with like serious questions.
Good.
And yeah, I'm just rather surprised. I think the only like unserious question is the most difficult one to answer.
Yeah.
From Latham was, what do you think is the weakest part of your dissertation?
Oh my God.
Yeah.
Now I wonder if Walter and Keith are gonna ask stuff like that. Oh my
I
God.
don't know. They may have seeded their minds with things to ask.
That's great. Interesting. Did you get a lot of questions from the stack or?
Oh yeah,
You
yeah.
did.
So I kind of I focused in on like maybe 10 papers from the stack that had things to do with my proposal.
Good.
And I
Yeah.
kind of dropped those names in early. And I
Hi.
think
How you
that
doing?
neutralized a lot of further
See you guys.
questions from him.
Fine, right?
Yeah.
Yeah.
So went on the offensive.
Yeah.
Good.
It's an exciting
Yeah.
day, isn't it?
Surprising,
Like,
successful maybe.
maybe. Then you squat. Then just kick right.
She's not wrong.
Up my grab. Just kick back and say, go there, go here. You know.
Well, we're certainly gonna miss coming to Kansas.
Yeah.
I could live in here.
Yeah.
Yeah.
You know, my dad just had knee surgery and like he always wanted to come golf Culvert Hills one
Yeah.
time. Yeah. He's just like, man, I gotta come down and golf.
You know, I actually have 2 free passes right now that expire at the end of the year.
Oh, shoot.
Well, today would be the day to be doing
I
it.
Yeah,
know, let's-
yeah.
Well,
Just-
let's go.
all right. Perfect.
Let's go. I'd be happy with that.
Yes.
Wow, look at this.
I know.
Yeah, they went all out.
My
So,
goodness.
so I just retired from teaching after 35 years.
Yeah.
And we have renovated this big old family barn. Built in the 1870s.
Yeah.
My parents are, my grandma 80, 81. They built this one when
I
we
don't
had
recall
center
that.
kind of base. But I like to do just some small, like generous combos or just use the barn
Yeah.
for something.
Yeah.
So-
I mean, like you could put
and I
150
love
people
to-
in.
I love to entertain.
Well,
Yeah.
This is you, huh?
Yeah, this is her wheelhouse.
No gosh.
Yeah.
You know,
I don't know much about
I gotta tell you, carrot cakes are my
Oh.
absolute favorite. I didn't notice.
I didn't realize that. No, I do like carrot cake.
You ask anybody in the family.
And so I'm finding this time that I didn't tell her that.
Yeah,
Well,
Lubbock,
it was like besides
Yeah.
80 yards.
Lubbock is
So I started to go through some photos
it's a
and found
difficult place.
some other pictures of Elizabeth when she was younger.
I love, you have the one, where's my favorite one?
Yeah,
This
it's
one.
really far out.
Oh, yeah.
I think the t-shirt, you know, that's like classic mid-90s, like a
Most
little
ice
cat
cream
with
is
Yeah,
like-
cowboys.
That's, yeah.
I think it's the, it's that little look, that little twinkle in your
Oh,
eye.
Yeah.
I'm a little bit troubled.
Yeah. You're not wrong. And then this is my uncle, this is my advisor, Keith.
Hi, nice to meet you. Nice to meet you. Yeah, like, we make it. Yeah, right on. Yeah, it's great to have some support.
No, because then it's only
Yeah.
that.
It's
Well, it's
surprising.
almost over now. you know.
Yeah, it's a-
Yeah. Yeah. Yeah.
You care too much.
What do you do? Okay. All right.
Keep it contained up there.
Yeah. Yeah. Okay. The glorious species itself. Yeah. Cool. Yeah, fun. Yeah. How long you guys in town for? Yeah. Hopefully, you know, enjoy the short, yeah, time, so.
Oh.
How long you drive to the park?
6 hours.
Oh, that's not bad. Usually like letting everybody get their fill. I don't want to like eat something, but I'm gonna take
Yeah. Might have nice beer.
Y'all could say, have you ever met graduate students? You actually think there's something wrong? Yeah.
I don't know.
They'll be filling their pockets with everything.
Tomorrow morning, man. Sounds good. Okay.
Let's see here. Oh, we got people coming in. All right. Hi, folks. Welcome in.
All right, we got Jeff.
Yep.
Good deal.
Hi, Liz.
Let's see here. All right. Say, Jeff, just as a sound check, can you hear us all right?
Yep, perfect.
Okay, awesome.
Are we using this mic or?
No, we're using my laptop. So if they have questions, I'm just going to repeat the question for folks in the audience. Yeah.
So if I have an opening thing, I can just plug it in there.
Mhm. Let's get that in like right now here. I've got, or yeah,
Well, we got plenty.
yeah. I've got, there's an open USB on this side. So, yeah.
Nipper. Well, those aren't for faculty, because otherwise this vagabond faculty will just show up for the food, even though they study plants or something like that, you know?
Plant people can eat.
Emily, congratulations.
He's already told me what my first question will be. Why do fish suck so much?
All right. All right. Thank you for letting me know about that planted question. That's good.
Always was
Good deal.
It was kind of a disappointing day. Rough.
Yeah, I think I was.
Okay, good, there's Tom.
Okay, great. Okay, hi, Tom. You good? Okay.
I'm good, Liz. Thanks.
Yes, yeah.
How about you?
We haven't heard from Peg, have we?
I have not. I think I need to turn. There we
Can you
go.
hear me okay?
I'm sorry, what was that?
Can you hear me okay?
No, I
No.
can't hear him. I can't hear him okay. Is that
I
something
hear
with
you.
my audio output, maybe? I
hear
couldn't
you
hear.
great. You sound wonderful, but.
There we go. Okay, let's see. Can you say something now? Can I hear you now?
Testing, 1, 2.
There we go.
3.
Perfect. Okay, got that figured out. Thank you so much.
Good thing.
And water used by grasses.
I got you.
It's too bad they didn't
Oops.
Walter, you gonna get enough fish?
Yeah, you can get all fished out today.
Not enough.
That's true.
Here,
That's true. Multiple trophic levels, just for you.
I made Peter talk about read today.
Good.
Oh, brutal. He made me listen to him.
Oxidized sugar.
That's pretty good. I think you got a good Zoom crowd probably.
Yeah.
Head on there?
My brother's on here. Let's see. We got Jeff. Yeah, Ted's here now. Okay, cool.
Okay.
Hey, Ted.
Head
Come on. I'm ready to
Awesome.
watch you rock it. Let's do this thing.
Let's do this.
Excellent. Got the committee here. Well, let's load up my intro here.
All right.
You're not sharing
I'm not
screen or
sharing.
anything?
Not yet.
COVID.
We can just ignore that.
Goldfish going all
It's the,
Cleaned up all the water and dropped.
oh, there it is.
Got it and I spilled it everywhere. It was like this.
What?
Just spilled it everywhere.
1 minute.
They didn't need to, like, smoking by.
What'd you get? The carrot cake?
It was so
Yeah. I got mine waiting for me.
This is gonna carry me to the afternoon.
Okay, good.
Perfect.
That's awesome.
That's what I was hoping for. It wasn't.
Well, I think it's 2:30, right?
Yeah.
All right, we might as well get started. Hopefully everybody online can hear me all right here. You guys are all just a little box right now. Man, it's a great pleasure that I get to introduce Liz Renner. She's going to defend her dissertation today. I will say that I want to first acknowledge the committee. Walter Dodds, John Blair are here in person. Ted Harris from KU is online. And the outside chair is Dr. Thomas Phillips. He's a professor in entomology, and he was appointed by the graduate school. Dr. Phillips is going to be, he's online, but he's here. So anyway, those are all the official people. The ground rules here are that all the folks on the committee will hold their questions until Liz is done and the general audience has been able to ask questions. Then we'll send everybody away, and then the committee can ask as many questions as they want. And so, like I said, this is great. Liz has been here. I've known Liz since 2016, I think, when she was an REU student. She came into the REU program and moved into the graduate program. So that happens to a lot of grad students. So she's a great example of a success story there. I took a quick look at, I've been out in the field, so I kind of had to throw this together last minute. First and foremost, of all my students, she does not study plants. So that's one of the best parts of her accolades. She has a BS degree from Augustana University. She's co-authored 4 peer-reviewed publications. She has a whole slew of other papers that she's working on finishing up once she defends here. She's given a ton of presentations in her time here at K-State and others. And before she got here, she was really accomplished coming out of Augustana. In the Division of Biology, she's won the Robel Award for Outstanding Ecology Graduate Student. She won the James Ackert Award for Outstanding Presentations. Within the Kansas chapter of the American Fisheries Society, she's been recognized by the Otto T. Meyer Frank Cross Student Scholarship and the Best Student Paper Award, as well as in the Dakota chapter of the ASS, so the Best Student Paper Award. So that's all the boring stuff. Liz has just been a lot of fun. No matter what you have her do, she's got a big grin on her face. If it's chopping through fish or busting through weeds to collect bugs. She does a lot of other cool attributes. She's a great artist. You ought to check it out sometime if you haven't seen some of her artwork. She draws wonderful fish pictures. She's proficient at field work. When she got here, I found one of her old goals. Every year we write goals, and she said, I want to break less equipment. But her mentor was a guy, Ben Neely from KDWP2, who breaks equipment like it's glass. And she learned from him. And she also speaks Spanish. So if you can interpret that, that's what Google. She speaks Spanish from Google. Anyway, Google translation.
Muy bien, Keith.
Yeah. But my favorite thing about Liz is her ability to overcome advisor-induced adversary or adversity. Sorry, she's been through a lot, and I just cherry picked a few things here. But Liz is just, it's been something new every minute, every hour, every year. You know, I sent her off to do all this field work in Western Kansas, driving hundreds of miles with a boat where the bearings haven't seen grease since the 1970s, since Walter was hired. I asked her to house sit, and I didn't tell her about the killer bird that lives in my house, and the bird tried to kill her. That's him. He's about 2 inches tall, but don't get me wrong. By the way, if anybody wants a bird, it's still alive, and it's yours if you want it. Just say the word. Her first field companion was Sky Hedden. If you know Sky, I don't need to say anything else. If you don't, maybe that says it. You know, I remember Liz won me over early on when I went down to the basement in the lab, and she was just chewing this kid out. I mean, she was letting him have it. This was this wonderful Hutton junior fisheries biologist program that the American Fisheries Society, where they bring a high school kid in and mentor him. This guy was a real piece of work. And he, like, for example, Eli reminded me that when he took data, he liked to write numbers on top of each other. So, so that's what the data sheets would look like over there. When you, anyway, Liz let him have it, and I just had a warm spot for Liz after seeing her tearing this kid a new one. I know she got familiar with this diesel-powered water sprayer. That was probably the biggest pain in the butt for her whole experiment. And then I sent her out west, and I don't know what happened there, but this picture sort of was kind of funny when she came. Somewhere during that trip, things got a little loopy, I guess. But anyway, with that, you know, Liz, it's been a pleasure to work with you, and I think we're all looking forward to seeing your presentation. So we'll just let you do whatever you can here. I'm gonna just pull that out.
Perfect. All right, let's see here. Share this. Okie dokie.
Hopefully I didn't crash the system.
No, I think we're good. All right, thanks for that introduction, Keith, and thank you all for joining us today, both in person and on Zoom. I'm so excited to be able to share my work here. And so before I get started, I wanted to just go ahead and acknowledge some folks that I really want to thank for getting me to this point. So first, I'll start with my advisor, Keith. So I've worked with Keith now, like you mentioned, since 2016 when I came in as an REU student. And you know that you've been in the same lab for a long time when you catch yourself over time saying and doing the same things as your advisor. So just for example, in recent years, you know, I've caught myself saying some Gidoisms, for example, like, you know, don't drop the bucket, or damn it all to hell, or all models are BS, and things like that. And for instance, for those of you that have ever received an email from Keith, you know that he has a tendency to use a lot of ellipses and kind of trail off at the end of emails sometimes. And as a graduate student, you know, sometimes that can be like really positive and open-ended, and other times it can be like confusingly ominous. So, but I have picked up on that. That's a habit that I've learned along the way. But just in general, I wanted to say, like, I am so grateful for Keith's mentoring and support. He's really helped me grow in confidence as a freshwater scientist. And yeah, I'm just so grateful that he's helped me learn and grow over the years. Next up, I want to thank my dissertation committee. So Walter, John, Tom, and Ted, I'm grateful for their support and mentoring along the way, and for helping me with my writing. Next up,
Hey Liz, I appreciate that. Thank you. But what I'm really trying to say is we can't see the slides you're presenting. We can only see the setup of the slides you're presenting on Zoom. It happened with Keith as well.
Oh, thank you.
So we just see, like, yeah.
You can only see me.
We can see you, and then we can see, like, when you first open PowerPoint, all the slides are on the left, and then we have your intro slide there, but we're not seeing the slides actually turn. Yeah.
Is this it now? Can you see that?
There you go.
Is that
Perfect.
it?
Thank you.
Okay.
Yeah,
Great.
yeah.
Glad we figured that out. Thanks for catching that. All right.
No worries. Thank you. Keep going.
Awesome. So next up, I wanted to acknowledge all of my lab mates, past and present, in the Gido lab. I've been really blessed with all of their support and encouragement. We've had a lot of fun together out in the field and in the office working on our code, and I wouldn't be where I am today without their support and help. Next up, oh gosh, that's not advancing now. Here we go. There we go. Next up, I want to acknowledge the support and the assistance of all of my amazing technicians and volunteers who made this process or this project possible. It takes a small army to study 12 small impoundments over the course of 4 years. And so this wouldn't have been possible without the help of so many talented undergrads that I've had the pleasure of working with. Then next up, I need to thank my amazing collaborators at the Kansas Department of Wildlife and Parks for making this project possible. I could count on them to drop everything and meet me at a boat ramp with a backup electrofishing boat or an Ekman dredge at a moment's notice. And so they all really went above and beyond to lend their time and talents to make this project possible. I also want to acknowledge Dave Hocus with the K-State Co-op unit for letting us use the co-op unit's electrofishing boat and storage space for the course of this project. And then I want to acknowledge the support of all of my friends in the division. I've been so blessed to make lifelong friends here at K-State who have really supported me along my journey here at K-State. And then I need to acknowledge the support of my fantastic family. It takes a village to raise an aquatic ecologist. And so I wouldn't have made it this far without their love and support. And they've really fostered my passions over time, supported me in all of my crazy pursuits. And I just want to acknowledge my dad wasn't able to make it today because he just had knee surgery, but I do have my special guests. My mom, Karen, and then my uncle Eric are here with us. And means so much to me that they could be here today. And then finally, I need to acknowledge my wonderful little research assistant, Banjo, for his support. He's been a constant companion here in Bushnell and throughout the pandemic. So he's helped me. And then with that, we'll go ahead and get started. So the title of my dissertation is a bit of a mouthful, but today I'll be sharing my work on disentangling the ecological role of gizzard shad in Kansas small impoundment food webs, changes in limnology, fish communities, and predator diets after rote node manipulation. Okay, so I'll start off this talk just with a brief introduction to gizzard shad ecology and what motivated us to study them in Kansas small impoundments. Then I'll get into our project objectives and discuss some of the characteristics of our study impoundments. And then I'll walk you through my first data chapter looking at associations between gizzard shad and different water quality parameters in Kansas small impoundments. Then in my second chapter, I'll cover our use of replicated low-dose rote node manipulations to study changes to different food web compartments after shad removal. And then my third data chapter explores variation in rates of largemouth bass piscivory as a function of shad abundance, seasonality, and ontogenetic changes. And then finally, I'll summarize my findings, kind of wrap things up, and I'll finish by taking your questions. All right, so let's get started. First, I wanted to introduce you to my focal organism. Many of you who have heard some of my talks in the department so far have already been introduced to the lovely gizzard shad, but for those of you that don't know, gizzard shad are a freshwater forage fish from the family Clupeidae that are closely related to herrings, anchovies, and menhaden. And so gizzard shad are a really unique fish. They tend to dominate reservoir fish communities and resist management efforts for a number of reasons. They have a really unique life history that makes them really successful members of reservoir food webs. And so gizzard shad can compete directly with age 0, so juvenile bass and bluegill, and indirectly impact sport fish by a variety of factors. So gizzard shad grow really rapidly, they reproduce very quickly, and then they're facultative omnivores. So they're really unique in that gizzard shad primarily are planktivorous, and so they can feed on zooplankton and phytoplankton, and they're filter feeders. But at times when those resources are limiting, they can switch to feeding on the decaying organic matter, benthic detritus, on the bottom of an impoundment. And so they're unique in a food web in that they're able to metabolize the microbes and decaying organic matter for energy. And so their ability to do that when they're feeding on that benthic detritus can resuspend inorganic nutrients into the water column and resuspend sediment, which can negatively impact water quality. And so they do have the potential to both directly and indirectly impact sport fish. And so there's been a considerable amount of research that's been done analyzing the relationships between gizzard shad abundance and different components of food web structure. So first and foremost, a number of studies have documented this positive relationship between gizzard shad abundance and increasing primary productivity. So higher concentrations of total nitrogen, phosphorus, and chlorophyll concentrations in reservoirs and small impoundments. At high gizzard shad abundance, there's also a pattern of a relationship between a reduction in total zooplankton biomass and a dampening of size structure towards smaller sizes of zooplankton. There's also this relationship, as I mentioned earlier, due to their negative impacts on juvenile sport fish, so that at higher gizzard shad abundance, we tend to see some lower abundances sometimes of bass, like largemouth bass and bluegill. And then finally, one study by Schaus et al. From 1997 found that because gizzard shad are resuspending nutrients into the water column, and they're excreting at a low nitrogen to phosphorus ratio, that under those conditions, that excretion could be providing a subsidy of increased phosphorus into the water column. And under those conditions, that would be favorable to an organism like a cyanobacteria that can fix nitrogen. And so there's that relationship between potentially higher shad abundance and higher blue-green algae abundance. So juvenile and adult gizzard shad are playing this role in providing potential nutrient subsidies to phytoplankton via that excretion of sediment-derived nutrients. So they're excreting and resuspending inorganic nutrients like phosphorus and nitrogen into the water column. And so that provides a nutrient subsidy to stimulate phytoplankton growth. And then this can potentially facilitate eutrophication in small impoundments. So then with that eutrophication, we don't understand as well how those influences on primary productivity are then influencing relative abundances of gizzard shad in these systems. So here is just a really oversimplified diagram of a typical Kansas small impoundment food web. And so as you can see, if you look over here, gizzard shad occupy this intermediate trophic level, and they kind of link the benthic with the pelagic food webs. So their impact on small impoundment food webs is bidirectional, or kind of middle out. So first, they're exerting top-down effects by directly influencing sport fish, since they serve as a prey resource for piscivores, and they're also competing for co-occurring juvenile fishes for limited zooplankton resources. But they're also exerting that bottom-up control by regulating nutrient cycling when they resuspend those inorganic nutrients into the water column, which can potentially fuel primary productivity. And so to summarize, gizzard shad are ubiquitous. They're found in reservoirs and small impoundments across a broad swath of the United States. And so they really are widely distributed, and they're still being intentionally stocked as supplemental forage in some reservoirs. And they're accidentally being introduced via flooding and bait bucket introductions by anglers in other places. And so their range is expanding. And there's a growing concern about their potential negative impacts to food webs and the associated management costs. And so we don't fully understand their role within small impoundment food webs. A lot of the past research that's been done on gizzard shad has been conducted in larger reservoirs, and at like Mike Vanny's work in Ohio reservoirs, and there's been work done across the Southeast and in Illinois reservoirs, but less is understood about their ecology within these smaller systems where there are smaller populations of open water piscivores like walleye or wipers. And the next few studies have examined the direct effects of gizzard shad on water quality or lower trophic levels using experimental lake manipulations. And so that kind of led us to our interest in this study. And so in 2016, a survey by our collaborators with the Kansas Department of Wildlife and Parks revealed that overabundant gizzard shad are becoming a growing management concern in Kansas small impoundments. And specifically, they cited concerns about potential negative impacts on sport fish growth rates and water quality issues. And so there's this growing interest in using a pesticide called rotenone at low doses to selectively remove gizzard shad from these food webs. But there are concerns about any detrimental impacts to other species, or concerns about what happens when you remove them from the food web. And so that prompted my collaborators, Ben Neely, Jeff Cook, and then Sean Lynott with the department to do this pilot study at Montgomery County State Fishing Lake. And so they went out in 2014 and they did baseline sampling, and then applied low-dose rotenone, and then evaluated the relative abundances, growth rates, and changes in body condition for both bass and bluegill after manipulation. And they found that gizzard shad reduction had a really minimal effect on these bluegill and bass populations. So they didn't see substantial substantial changes in these parameters over time. And so the growth of bass and bluegill improved just a little bit initially, but then declined in subsequent years. And they didn't really understand why. They thought potentially it could be due to compensatory mechanisms or, just kind of a mass response. So we wanted to get at a more mechanistic understanding of what gizzard shad are doing in these food webs and what's driving that. And so that leads us to my dissertation project. And so for my dissertation work, we kind of wanted to look at 3 different components of the ecology of gizzard shad in these systems. And so first, we wanted to analyze differences in water quality between impoundments across a range of gizzard shad relative abundances to look at their effects on nutrient dynamics and primary productivity. Then secondly, we wanted to evaluate any food web responses to selective shad removal with our replicated low-dose rotenone manipulations to see what happens when you take shad out of an ecosystem. And then finally, we wanted to characterize any differences in largemouth bass diet composition between impoundments with and without shad and see what was driving variation in these impoundments and whether or not it was related to gizzard shad abundance. And so for this study, we conducted this project in 12 small impoundments throughout the state of Kansas. And so here's a map of our study impoundments. All impoundments had similar fish communities typical of warm water reservoirs across the Midwest. And they all varied in size from about 17 surface acres to 300 surface acres. So they're pretty small impoundments. So we conducted this study across these impoundments, and we had a gradient at the start of the study, 4 impoundments without gizzard shad and 8 impoundments with gizzard shad. And so that brings us to my first chapter, looking at associations between shad relative abundances and limnological variables in these small impoundments. So as many of you are probably aware, cyanobacterial blooms present a growing public health risk in lakes and reservoirs across the United States. Here in Kansas, evidence is mounting that we're having a lot of blooms in our different federal reservoirs and some of these state fishing lakes and reservoirs. And so because benthic-feeding fishes like gizzard shad and common carp are potentially resuspending nutrients and sediment into the water column and subsidizing primary productivity, we wanted to understand, are these benthic-feeding fishes playing a role in subsidizing some of this growth? And we wanted to understand whether or not they could be contributing to this issue. So for my first chapter, we asked overall, are there any associations between gizzard shad abundances and the limnological characteristics in these impoundments? And so specifically, we anticipated that we would see higher total nitrogen and total phosphorus concentrations at higher gizzard shad abundances. And we also anticipated that we would see a reduction in water clarity with more gizzard shad. We anticipated that we would see reduced zooplankton densities with increasing gizzard shad abundance. And then specifically looking at the size structure within zooplankton populations, we anticipated that gizzard shad would be selectively feeding on larger bodied cladocerans compared to smaller bodied rotifers and copepods. And so we'd see lower densities of cladocerans and a greater dominance of rotifers and copepods with increasing shad abundance. And then finally, with respect to phytoplankton, we anticipated that we would see an increase in the total algal abundance at higher gizzard shad abundances. And that specifically due to that, their contributions to the N/P ratio, that we would see higher abundances of cyanobacteria at higher shad abundances. And so we hypothesized 2 mechanisms by which higher gizzard shad relative abundances could be leading to increased algal biomass. So first on the top here, we hypothesized that total nitrogen and total phosphorus would be positively associated with gizzard shad relative abundance, but that phosphorus would increase at a faster rate than nitrogen, resulting in a net decline in the N/P ratio with increasing shad relative abundance. And so these effects on nutrient availability should stimulate algal production, particularly of nitrogen fixing cyanobacteria because of that reduced N/P condition. And then secondly, on the bottom here, we anticipated that zooplanktivory, so the consumption of zooplankton by gizzard shad, should increase with shad abundance. And so resulting in both a shift towards smaller zooplankton taxa and then reduced overall rates of herbivory by zooplankton on phytoplankton, resulting in an overall increase in algal abundance. So for this project, we used a bit of a regression design. So we were looking at relationships between shad abundance and our response variables of interest. And so we sampled, like I mentioned, these 12 small impoundments with variable densities of gizzard shad. And so at the start of the study, we had 8 impoundments with shad and 4 impoundments without shad. And we sampled them over the course of 4 years from 2017 to 2020. And so we collected data for different limnological parameters at 3 stations from each impoundment in June, August, and October over the course of those 4 years. So stations were positioned in limnetic habitats in the riverine zones near an impoundment's inflow up lake, and then in mid-lake transition zones in the transition zone, and then in lacustrine zones close to an impoundment's dam. And so we collected samples at each of these stations that were used to quantify water chemistry parameters. So we were looking at total nitrogen and total phosphorus. We also we did vertical and horizontal zooplankton tows to estimate zooplankton densities and composition. And then we also collected surface water pelagic algae grab samples to estimate phytoplankton abundance and composition. And then we also needed to evaluate gizzard shad densities and determine whether or not there was actually a relative abundance gradient that existed in these fall impoundments. And so to do this, we use standardized boat electrofishing surveys to quantify gizzard shad relative abundance over time. For our laboratory methods, we thawed out our pelagic water grab samples and analyzed them for total nitrogen and total phosphorus with the rapid flow analyzer through the K-State soil testing lab. And then for our phytoplankton samples, we settled out our samples and took photos using a camera mounted to an inverted microscope. So we were able to create this photo library of our phytoplankton samples. And then we superimposed these counting grids basically to get a metric of coarse metric of algal aerial abundance, basically. So we counted the number of grids occupied by each taxa and used that as a coarse estimate of our algal abundance. For our statistical analyses, we fit these linear mixed effects models to look at relationships between shad abundance and our dependent variables. So our fixed effects, we had shad relative abundance as catch per unit effort electrofishing, sampling month, and then that interaction between abundance and month. And then our random effect of impoundment to account for the variation across impoundments. And so for any of these relationships with significant interactions, we did post hoc models to look at them at a finer resolution. And then we wanted to compare the marginal R squared, so the variance explained by those fixed effects only, to our conditional R squared, so the variance explained by both the fixed and the random effects, to look at that for all of our models. All right, so getting into our results for total nitrogen, we'll start off with our nutrients. And so we found that there was a significant interaction between shad abundance and month. So there was a positive increasing relationship between shad abundance and total nitrogen concentration. So over here on the x-axis, we have shad CPUE, and then we have log transformed total nitrogen. And so you can see that there was a positive relationship between TN and shad abundance in August, but this negative relationship in October. So there was a month effect. Next up, looking at total phosphorus, we also saw a significant interaction between shad relative abundance for total phosphorus. So as you can see here, phosphorus is increasing with increasing gizzard shad relative abundance, but this relationship was stronger in August and in June than October, which looks kind of weird from those slopes of that lines, but we did see a stronger relationship in the first 2 months rather compared to October. Next up, looking at N/P ratios, we did see a significant interaction between shad relative abundance and month for N/P. So we did not see any significant changes across shad abundances for June or August, but we did see significant declines in October with increasing shad relative abundance. So you can see that increasing over here. So N/P is declining at higher shad relative abundances. Next up, with respect to phytoplankton, we did not see a relationship between shad abundance and phytoplankton abundance, contrary to our hypotheses. So we did not find any interaction between shad abundance and month. And so up here on the upper left-hand corner, we have our blue-green algae abundance, so the cyanobacteria, there's no relationship. And then we have green algae, diatoms, and then total algal abundance in the bottom right-hand corner. And so there was no relationship with any of these variables. Now getting into zooplankton densities. So generally total zooplankton densities decreased at higher gizzard shad abundance. And this pattern was consistent across months. And so up here on the upper left-hand corner, we have our cladoceran densities. And so all of these are square root transformed here. So we have declining cladoceran densities at higher shad abundance, along with calanoid and cyclopoid copepods up top. And then on the bottom, we have rotifers. The relationship was not significant for copepod nauplii, but it was significant. There was a decline with increasing shad abundance for total zooplankton density. Okay, so to summarize, at higher gizzard shad abundances, we saw higher TN and TP concentrations. We also saw that at higher shad abundances, we had lower overall total zooplankton densities. And so there was no relationship between shad abundance or total phytoplankton or cyanobacterial abundance. And so this positive relationship between shad abundance and nutrient concentrations indicates the potential for a bottom-up effect of shad on primary producers. And in contrast, that reduction in zooplankton densities at higher shad abundances is indicating a potential for top-down effects through that reduced algal grazing by zooplankton. And so we also failed to support that hypothesis that there'd be a relationship between shad abundance and algal abundance or cyanobacterial abundance. There we go. And so we also saw that at lower NDP ratios, we saw lower NDP ratios at higher shad abundances, and that pattern was consistent with findings by Schaus et al. And so these bidirectional effects on zooplankton density and nutrient concentrations were pretty consistent with the literature. So we did find evidence that this has happened in other systems. And so overall, our observations that gizzard shad are influencing these nutrients in zooplankton suggest that they do have the potential to contribute to harmful algal blooms, but they're not significantly altering these phytoplankton communities. So that's kind of an interesting result. And so shad might be weakly stimulating eutrophication in small impoundments, but the mixing regimes and the watershed level factors like land use might be playing a larger role in driving these patterns in nutrients and influencing potential cyanobacterial blooms. And so it's worth noting, sampling just 3 snapshots in time over the course of a season might have been too limited for us to detect any kind of intermittent or fleeting gizzard shad effects at different abundances. But it's worth noting, so the ratio of marginal to conditional R squared in our mixed models was large. So that suggests that the variation among impoundments was more pronounced than the variation or the fixed effects of the shad relative abundance and month. And so therefore, identifying that the role of gizzard shad across this range is a little bit confounded by these watershed level factors. And so Kansas small impoundments are highly variable across the landscape. And so past work that my collaborators, Ben Neely, and that we've done demonstrated that these impoundments are really variable by watershed land use, size and morphology, the mixing regime. So whether or not they're mixing twice a year, just a typical dimictic lake, or they're polymictic and mixing constantly whenever there's intense wind disturbance, and that can influence dynamics within these systems. And then they also vary greatly in terms of total macrophyte cover, which has important implications for juvenile fish communities. And so watershed land use upstream of these impoundments could have a confounding effect on phytoplankton dynamics. So we found that the percent agriculture in the watershed upstream of our impoundments was correlated with total nitrogen. And just in general, so we found both from our study and from what I found in the literature that the evidence for strong gizzard shad effects on phytoplankton is mixed. And a lot of these responses are highly context dependent on the underlying biomass and size structure of gizzard shad in these systems. And then interestingly, I also found that there's kind of a variation across the board in how we approach modeling gizzard shad relative abundance. So some studies treat gizzard shad as drivers of some of these parameters, whereas other studies are treating them as a symptom of eutrophic conditions. And so they model them as response variables. So that's kind of a question of what came first, chicken or the egg question, the high gizzard shad abundance or the underlying eutrophication issue in the impoundment. And so that makes things more difficult to disentangle for sure. But in general, that led us to want to look at these relationships. And we looked at all of these correlations, kind of looking at associations over time, but that inspired us to do an experimental manipulation to really parse out these relationships. Which brings me to my next chapter. So after examining these in a regression design, we wanted to test the experimental removal of gizzard shad with replicated root node manipulations. So in this chapter, I'll focus on characterizing changes to lentic food webs in response to low-dose root node manipulations. So as I mentioned in my introductory material, low-dose rotenone manipulations have been explored as one possible method to selectively reduce or eliminate gizzard shad that are overabundant in different impoundment systems. And so unfortunately, these systems these methods result in improvements to water quality and sport fish growth rates, but any kind of responses and potential changes to body condition and relative abundances are often limited or non-existent and really fleeting. And so we don't fully understand why, but again, it could be due to compensatory mechanisms or other mechanisms we don't fully understand. So that led us to lay out some objectives and questions for my second chapter. So first, we wanted to look at the effect of selective shad removal just in general on all of these food web components before and after manipulations. And so specifically, we wanted to test for changes in those limnological parameters I discussed in chapter 1 and compare them to control lakes with similar fish communities and parameters to see what happens when you remove shad. And same thing, we wanted to test for changes to fish community dynamics with a specific emphasis at looking at changes to largemouth bass and bluegill relative abundances and growth rates and whether or not they change after shad removal. And so now I want you to think back to my first chapter and those hypothesized mechanisms, because we're going to flip things in our mind and kind of think opposite from the last chapter. So in this chapter, we hypothesized that shad removal would elicit reductions in pelagic nutrient concentrations. And because that benthic foraging by gizzard shad and organic detritus is subsidizing primary production. So you take them out of the system, and in theory, that subsidization should decrease. And so we anticipate that when you remove shad, there would be greater reductions in total phosphorus than total nitrogen, which will increase the N/P ratio and subsequently lead to a reduction or potentially less favorable conditions for cyanobacteria. And then next, we hypothesized two mechanisms which would lead to, for gizzard shad removal, their effects on zooplankton. And so we hypothesized that zooplanktivory would decrease following gizzard shad removal, and that the lack of that zooplankton or zooplanktivory on feeding on zooplankton communities would lead to an overall increase in biomass and size structure of zooplankton. And so that increased herbivory of zooplankton on phytoplankton communities would result in overall lower abundances of phytoplankton. And then for fish community responses, we predicted that the relative abundances of planktivorous fishes such as bluegill and largemouth bass, young of year largemouth bass, would increase following shad removal because of that reduced interspecific competition for zooplankton resources. And so for this project, we used an experimental design that was a Baki design, so before after control impact design with 2 treatment groups. And so we identified 8 of those impoundments with gizzard shad communities. And so we divided them into 4 treatment impoundments and 4 control impoundments. And so we did 2 years of baseline sampling before rotenone manipulations. And then, so we collected all these baseline measurements, and then we applied rotenone in December of 2018, and then tracked these food web variables for 2 years post-treatment. Unfortunately, things happen, and field experiments in ecology rarely go as we plan. And so unfortunately, our rotenone treatments were unsuccessful in 2 of the 4 treatment lakes, and we did have gizzard shad rebound in those lakes. And then we also had a partial dam collapse in one of our control impoundments. So that left us with a reduced sample size of 5 impoundments, so 2 treatment impoundments and 3 controls. And so here are just some photos from that rotenone manipulation in December of 2018. So we tried in December to pump chemical at 5.5 to 7.5 parts per billion to selectively target gizzard shad while trying to reduce incidental mortality of bass and bluegill. And so once we applied chemical, we came back afterwards to try to quantify mortalities. And so this is a photo from Shawnee State Fishing Lake 2 days after the application. As you can see, there are a lot of dead gizzard shad in here, but also some dead crappie. So we did have incidental mortality. And unfortunately, so we tried to follow up and quantify the mortality here, but the ice cover over the lakes and wind and mixing made it very difficult for us to effectively quantify them because a lot of carcasses just sank to the bottom of the impoundment under the ice. So that was unfortunate in terms of quantifying mortality. So for this chapter, for our field methods, we did the same methods as I discussed previously for limnology in my first chapter. But for our field work, we conducted spring daytime boat electrofishing to estimate largemouth bass abundance during that spring spawning period. We also used trap netting to estimate bluegill abundance over time. We were interested in changes in fish growth rates over time. And so we wanted to calculate length at age and compare length at age before and after rotenone manipulation for both bass and bluegill. And so we pulled otoliths, which are these calcified structures in fish ears, kind of like an ear bone, and you can age them like tree rings. And so each of those little lines on there, those rings are called annuli, and the relative spacing between those annuli is a coarse estimate of their growth rates, is related to their total length. And so that's one way to evaluate changes in fish growth from year to year. And so we mounted those otoliths, sectioned them, and aged them via consensus reading to look at changes at length at age for these bass and bluegill. For statistical analyses, we fit linear models with our Baki design to look at how shad eradication affected these abiotic and biotic variables. And so we assumed, our design assumes that there's interannual variation within these impoundments. And so we're trying to focus on the relative differences in responses between our treatments and our controls. In this model, we had our fixed effects of impoundment treatment, so rotenone versus control, and then time period, so before rotenone and after rotenone. And so if for any of these interactions, we found a significant interaction at 0.1 alpha, it was indicating a significant treatment effect. So to get started looking at nutrient dynamics, we found that there was a significant interaction between treatment and time period for both total nitrogen and total phosphorus. So over here on the left, we see that total nitrogen decreased 41% after rotenone compared to 14% in the controls. And then on the left over there, or excuse me, on the right, we see total phosphorus decreased 37% in the rotenone treatments after shad removal compared to a reduction in the controls. So we did see reductions in total nitrogen and total phosphorus following shad removal. Next up, for our phytoplankton abundances, we did not observe any differences in total phytoplankton abundance after rotenone, and we did not see any changes in cyanobacterial abundance either. So as you can see, there was a lot of variation there, but no substantial effect of shad removal. All right, then getting on to zooplankton densities. Overall, we didn't see a change in total zooplankton densities or the densities of most of these taxa after rotenone manipulation. We did see a 6% decrease in calanoid copepod densities following rotenone manipulation in those treatment impoundments, but that was the only taxa of zooplankton that responded. And then finally, looking at juvenile bass abundance, we saw an increase in 16% in largemouth bass abundance for bass less than 150 millimeters after rotenone manipulation, but we did not see a significant change in adult bass abundance following rotenone manipulation. Then getting into bluegill abundance, we saw adult bluegill abundance here on the left decline 16% after rotenone manipulation. And then juvenile bluegill abundance on the right decreased 84% in response to rotenone manipulation. We also evaluated, as I mentioned earlier, those changes to length at age for bluegill and bass, and we didn't find any substantial changes in growth rates for bass or bluegill. And we were also limited by our sample size with age 0 bass. All right, so in general, you're probably wondering, well, did rotenone work? Was it a solution? Well, first off, so we saw that we had pretty limited responses. We did see responses in nitrogen and phosphorus, and we saw a response in our zooplankton densities. But in general, we didn't see any pronounced changes for phytoplankton communities. And so in general, it's worth noting our baseline gizzard shad relative abundances might have been too low to necessitate high rates of detritivory. And so if there are too few, I guess, lower abundance of shad, there's reduced inter and intra-specific competition for those zooplankton resources. So maybe they weren't needing to forage on the benthic detritus. And so if they weren't benthic foraging as much, that wouldn't have been resuspending nutrients into the water column and subsidizing that food web as much. So that it played a role in here. And then just in general, these rotenone manipulations are really expensive. They're difficult, labor and time intensive to execute. And so as illustrated by 2 of our 4 treatments didn't work. And so that makes things difficult. And just in general, you can kind of think of them as they might be temporary bandages for dealing with overabundant shad populations. And they're not even effective at that. And so they're just one potential tool in our toolbox as fisheries managers. But I would argue that multi-pronged approaches and in these systems and repeated manipulations combining both rotenone manipulations and maybe removal via gill netting might be necessary to control overabundant shad populations over time. All right, and now that brings us to chapter 3. So I'm going to share some of my work looking at different drivers of variation in largemouth bass diets and piscivory in these impoundments, and looking at changes to piscivory before and after shad removal. All right, so we know that there's a variation in the overall prey base of Kansas small impoundments. And this variation could be driven by the presence or absence of gizzard shad as a prey fish, as compared to other prey fishes like golden shiner, bluegill, green sunfish. And so these prey fish populations and their composition is directly influencing the abundances and the growth rates, the prey availability for predators that are recreationally important, like largemouth bass. And so in this chapter, just in general, I wanted to work with our collaborators to first just develop a general description of largemouth bass diets in these impoundments, and then look at relationships between comparing the frequency of occurrence of gizzard shad in these diets to that of other fish prey to see whether or not bass are actually foraging on gizzard shad in these systems. Next up, we wanted to evaluate associations between the frequency of occurrence of piscivory in bass diets and gizzard shad abundance, bass length, and sampling month to see if there were any ontogenetic or seasonal patterns. And then we wanted to determine whether selective shad removal was influencing piscivory in bass diets. And so first, we wanted to see, is piscivory in bass diets associated with shad relative abundance? So we anticipated that we would see a greater prevalence of piscivory in bass diets at higher shad abundances. We also wanted to know, do rates of bass piscivory vary predictably across months? And so we anticipated that we would see the highest frequency of occurrence of fish in August in conjunction with the highest water temperatures and metabolic rates. And then finally, we wanted to look at whether or not piscivory varies with largemouth bass length. And we anticipated that we would see the highest frequency of occurrence of fish in adult bass, the largest size classes of bass, greater than 250 millimeters. And then with respect to the rotenone manipulations, we just wanted to know, does selective gizzard shad removal affect largemouth bass piscivory? And we anticipated, because we're removing a potential source of prey from the food web, we anticipated that we'd see reductions in rates of piscivory. And so for this chapter, we collected fish diets from largemouth bass via daytime shoreline electrofishing at our 12 different impoundments over the course of 4 years. And so we collected adult diets in the field, and then we also did seining in littoral habitats to collect juveniles at each of 3 sampling stations. So similar to my limnology chapter, we were sampling upstream, kind of mid-impoundment and in littoral habitats down by the dam. So we did this in June through October of 2017 to 2020. And then just in general, when we brought these diets back to the lab, we wanted to analyze each fish's gut contents under a dissecting microscope and identify them down to the lowest possible taxonomic resolution. And then we assigned these prey items to 1 of 14 overarching prey types and calculated the frequency of occurrence of these prey items within bass size classes, and then month for each sampling period, and then across sampling periods. And so for this component, we fit a generalized linear mixed effects model to look at that relationship between shad abundance and these components of bass diets. And so we have the fixed effects of shad abundance, month, and length class, and then the random effect of impoundment. Let's see, we also went ahead and fit a linear model using our Baki design to assess relationships between shad abundance and largemouth bass piscivory, similar to what we did in chapter 2. And so we had the fixed effects of impoundment treatment, so control versus rotenone, and time period, so before and after rotenone. So what are largemouth bass eating in these small impoundments? I'm glad you asked. We found, first, looking at relationships with bass length, we did see that there was a variation in these bass diets with bass length class. So just to orient you, on the y-axis, we have mean frequency of occurrence for these different diet items. So a value of 0 would be that item did not occur in any of the diets within a sample, and a value of 1 would mean that every fish in that sample had that item in their diet. And so we saw that the frequency of occurrence of fish was highest at these intermediate length classes. So in the middle here, this range of 150 to 250 millimeter bass. And then looking at adult bass, we were surprised to find that bass greater than 250 millimeters were eating crayfish, even slightly more than they were eating fish. And so that was something we didn't anticipate seeing. And then fish abundance, we found was not associated with gizzard shad abundance. So the frequency of occurrence of piscivory was not associated with gizzard shad. And our regression model indicated, again, that length class was the single greatest factor influencing piscivory in bass diets. And it was highest at these intermediate length classes. Then looking at the component of seasonality within these diets, we did not find any differences in piscivory across months. So there's pretty high diet similarity for these bass diets between June, August, and October. And then narrowing things in scope, just zooming in at the fish prey and the composition of fish prey for largemouth bass. On this figure over here, you can see we've got the count of stomach items in terms of occurrences of these different items. And then we've got in descending order of frequency of occurrence. You can see that the vast majority of the fish prey that we found were either too digested or not able to be identified positively down beyond just this is fish matter. But we also saw then that bluegill and unidentifiable lepomis, so sunfishes like bluegill and green sunfish, we weren't able to get down beyond that level of resolution. And then the third most frequently occurring fish prey in these diets was actually really surprising. It was cannibalism. So we saw a lot of juvenile largemouth bass that were in these diets. And then the fifth most frequently occurring item was gizzard shad. And then beyond that, we had some crappies and other rarer fishes within that. So in general, largemouth bass don't appear to be preferentially foraging for gizzard shad, but they are using them as a potential prey resource. But bluegill and other lepomis species seem to be playing a much larger role. And then with respect to our rote node manipulation, we did not see any changes in the rates of piscivory in bass diets in response to rote node manipulation. So didn't see a response there. And so in general, bass diets in these Kansas small impoundments were dominated by fish prey, crayfish, and then macroinvertebrates across length categories and months. Of the fish prey consumed, gizzard shad are only a small component. They're consuming more bluegill and sunfishes. And our regression model indicated that bass length class was a single greatest factor influencing piscivory. And that piscivory, though, was greatest at intermediate size classes instead of the largest size classes. And so this is kind of intuitive, right? So predator-prey interactions among fishes are generally size structured because a bass can't swallow and digest anything, but it can't fit its mouth around, right? So bass size is, it makes sense that it's related closely to the ability to consume fish prey. All right, and so now to kind of wrap things up and summarize all of this. So in chapter 1, we saw that high shad abundances were associated with lower total zooplankton densities, higher nutrient concentrations, but we didn't see that relationship between shad abundance and phytoplankton abundances. In chapter 2, I demonstrated that replicated gizzard shad removals with rotenone reduced total nitrogen and total phosphorus concentrations. And we saw increases in juvenile bass abundance, but reductions in bluegill abundances. But we did not see any effective shad removal on phytoplankton communities. And then in my third chapter, I demonstrated how gizzard shad are a prey resource for largemouth bass, but they don't seem to be the most important prey resource. So bass in these systems are eating sunfishes, and they're also cannibalizing their own young, and they're consuming crayfish, and those are the most dominant components. And so just to leave you with some take-home messages, at high abundances, gizzard shad appear to have both meaningful top-down effects on zooplankton communities through their grazing, and as well as bottom-up effects on fish communities via that competition and predation and their resulting effects on nutrient concentrations in these systems. But so their ecosystem effects in general appear to be substantial enough to merit monitoring and merit more management attention, but they might not be the most ecologically pressing concern in these systems. So small impoundments across the Great Plains are getting hit by many ecosystem stressors. And so one of them, we all are aware that ecosystems are being affected by climate change, and this has important implications for interspecific interactions. Invasive species introductions are influencing these interactions between species within these reservoir food webs. Eutrophication and related harmful algal blooms are becoming prevalent within these systems. And so just at a landscape scale, these are ongoing threats to these ecosystems. And, you know, associated land use change from land use, different agriculture in the watershed and urbanization are influencing nutrient dynamics within these systems. And then just in general, when you build a dam, over time, a reservoir ages and sediments in. And so you kind of got that inevitable march towards a reduction in productivity in an impoundment. And so that's something that needs to be counteracted in these systems. And then we have the negative impacts of pollution, chemicals, and increasing prevalence of microplastics affecting these food webs. And then finally, we just have a lot of habitat degradation associated with that sedimentation and changes over time. And so all of these kind of combine to create this massive problem in these systems, right? It's difficult to be able to mitigate against all of these factors working in concert. And so I would argue that while gizzard shad are playing a role in potentially influencing nutrient dynamics within these systems, they might not necessarily be the scapegoat or the right thing to point to in terms of what's driving patterns within these systems. So why should we devote more attention and resources to gizzard shad? I, again, would argue, and I hope that my dissertation work has demonstrated that understanding shad ecology in these small impoundments might better facilitate improved management outcomes in a time of unprecedented ecological change. And with that, I'd like to thank you all for your time, and I'd be delighted to take any questions.
Question?
Yes.
Any questions?
So when I think fish ecology, I also think dumpster fire. So that really resonated with me. So I might have missed it, but why is rotenone so good at killing shad, but not all the other fish that are in the lake?
Right. So thank you for that question. So rotenone it inhibits cellular respiration and it gets absorbed across gill filaments. And so it's not fully understood why rotenone affects some species more so than others, but some have hypothesized that it's the relative ratio of gill filament, gill mass to body mass that could be influencing that. And so a lot of the planktivorous fishes like a gizzard shad or a common carp seem to have higher mortality at the same doses of rotenone, same concentrations compared to other fishes like bluegill or bass. Yeah. Thanks.
Make sure you repeat the
Yeah. Oh, good point. Okay. Yeah.
First of all, great job, Liz.
Thank you.
So when I saw that Kansas map, I noticed how these impoundments are all over the state. So there's this really strong precipitation gradient happening. So in thinking that this precipitation gradient could be affecting heat limnological parameters, do you think that precipitation or differences affects these top-down, bottom-up feedbacks?
Absolutely. Yeah. Thank you. So the question for the folks watching on Zoom is a question about how the precipitation gradient from going from east to west across Kansas could be influencing some of these dynamics. And that's a great question. So I kind of mentioned how mixing regime in these systems could be making it more difficult for us to kind of parse out what's happening to these nutrient dynamics and phytoplankton communities. But yeah, I would say out in Western Kansas, where you have less intense rain events or just a reduced precipitation annually compared to Eastern Kansas, that the disrupting influence of a rain event, I guess that seasonality would be, it's more regular of an occurrence in Eastern impoundments like Neosho State Fishing Lake or Douglas State Fishing Lake. And so we wouldn't see that kind of influence as frequently out in our Western impoundments like Scott State Fishing Lake or Clark State Fishing Lake. However, just because it's not happening as frequently, sometimes with really intense events, that might mean that the magnitude of the actual impact of that pulse of potential nutrients in the system could be more intense and make more of a difference on disrupting stratification or influencing these dynamics in the Western impoundments than the Eastern ones. So that's a great question and a great avenue to pursue. Thanks. Yes.
That hit the precip gradient, so I'll hit the nutrient gradient. So it looked like most of the lakes you were on looked pretty goofy with a lot of eutrophication going on. So I was wondering whether you thought that the effects of shad introduction and increased abundance might be stronger in more oligotrophic lakes, starting that, bringing up all those nutrients might be more of a factor in these more quote unquote pristine lakes.
Yeah, that's a great question. So I guess, oh, sorry. So the question is about, just whether or not the underlying trophic states of these impoundments is influencing maybe the magnitude of the changes. So comparing an oligotrophic system to a eutrophic system when you have a gizzard shad introduction. And that's a great question, because I guess I didn't talk too much about it in this defense talk, but we did have 2 impoundments without shad. I'd say both of them more so Cowley State Fishing Lake than Douglas in the eastern part of the state. Neither had shad at the beginning of the study, but were introduced and are now at pretty high biomass. They've really taken off early on in these impoundments. And so, yeah, that's a great avenue of exploration. I think that I do think that in a system where there's, lower overall nutrient concentrations, having that impact of the resuspension of nutrients and the stimulation of production, they could have a larger role in a oligotrophic system than a eutrophic one. Thanks. I'm going to check here just to make sure that we have, let's see, got any questions in the chat here? Okay, I don't think so. Anybody else have any questions? Yes.
If big fish aren't eating the shad in impoundments, what's limiting them?
That's a great question. So the question is, if large predators aren't consuming gizzard, shad, and these impoundments, what is limiting them? And I would anticipate just availability of zooplankton and benthic detritus, but less so benthic detritus because there's just an abundance of it. So I'm guessing it's more the limitation of zooplankton resources, and they're competing with both for habitat and for food resources with co-occurring juvenile fishes. But yeah, you're right. So in larger reservoir systems, there are more open water piscivores like a wiper or a walleye that are able to consume gizzard shad, and they might be able to keep those populations in check more. So yeah, in small impoundments, the reason they can become so overabundant is because there isn't a top predator that's really regulating them. I will say that with my diet analysis, I didn't just do bass. I was looking at diets across the food web, and so we did process a lot of channel catfish and especially flathead catfish diets, and flathead catfish eat gizzard shad more often than largemouth bass do. So they're really the only predator that could potentially keep them in check. But they're, sawguy and flathead, I was actually surprised that those piscivores weren't consuming gizzard shad as often as I expected to see them. Thanks. All right, if there aren't any more questions. Thank you so much, everyone.
Thanks everyone for coming. Why don't we log off here?
How do I don't know why all of a sudden the meeting has stopped, but I need to stop and share. Thanks so much.
Yeah, you did great.
Thank you. Okay,
So,
let me see where this out here.
not sure.
I'm just like trying to stop the share.
Can't get a.
Screen sharing system work now. Remote control. Something's going on with my trackpad here. Huh. Okay, I'm just gonna temporarily disconnect this. There we go.
There we go.
That was weird. Okay. Thanks, everybody. Thank you all for coming. Let's see
Okay.
here. Okay. Thanks. Okay. There we
So,
go. Okay.
Tom and
And Ted's still here.
Ted
Yep, and Jeff's
and
still
Jeff.
on.
Hey, guys, there's another monitor with a better camera for a discussion than this laptop. So if y'all log out, I will send emails. I'll send an email with a new link to everybody, okay? Does that sound good? Okay, good.
Cool.
All right.
All right, so I'm gonna go ahead and,
Does this need to go into here?
I think so, yes. I'm gonna go ahead and end this recording if I can.